Earlier in this unit, we talked about the 3 levels of protein structure. Primary structure is all about the amino acid side chains and the order of amino acids within a polypeptide chain. The secondary structure are our alpha helices and beta sheets. And these, we learned, were held together by hydrogen bonding within the backbone. So BB here for backbone. Okay, and the third level, which is really we're going to kind of focus on in this video, is our tertiary structure. And this is the 3-dimensional structure of the protein, which involves interactions between secondary structural elements. So it involves the interaction between alpha helices and beta sheets. And since the backbone of the polypeptide chain is tied up as secondary structure, right? So the backbone is no longer reactive. So the forces that stabilize this 3-dimensional structure must be the side chains of our amino acids within the polypeptide chain. So that's what we really want to focus in on today. Okay, and just to kind of give this, I guess, a little bit of a pictorial view. If we focus in on our this cartoon diagram of a polypeptide chain we have up here, we see that as we start at the N terminus, the first secondary structure element we run into is a beta sheet, and the second one is also going to be part of a beta sheet. The third and fourth are alpha helices, and the fifth and sixth here are also going to be part of beta sheets. Okay, so if we think about how these secondary structural elements might come together to form a tertiary structure. So we start with the N terminus. We first proceed through our first strand here that will be part of a beta sheet, and we see that the second secondary structural feature is also going to be part of a beta sheet. So maybe we form this anti-parallel beta sheet here. Right? As we talked about, this is going to be stabilized through this hydrogen bonding between the carbon, the carbonyls and the nitrogen within the polypeptide backbone. Okay, the next secondary structural feature we see is this alpha helix followed by another alpha helix. So perhaps this alpha helix could maybe sit on top of our beta sheet here, right? So we have this interaction then between our beta sheet and our alpha helix that must be stabilized by side chain chemistry. The next secondary structural feature is another alpha helix, and maybe it's sitting out over here. And then finally we have our last beta sheet, which perhaps it's sitting something like this. Right, so I'm making up this structure as I go, but what I'm hoping that makes sense to you is that when this structure compacts into the structure that we know it needs to give it its function, there has to be interactions between these secondary structural elements that exist in the polypeptide backbone. And these need to be held together by interactions with side chains. Okay, so then without going into a lot of detail about every single intermolecular force that can exist between side chains, let's instead kind of focus on the overall big picture of tertiary protein structure. So what ends up happening, the reason these proteins fold into the structures they do is because they're trying to take all of the hydrophobic amino acids, all the hydrophobic side chains, and shove them together into the core of the protein. So this, what I'm drawing here, is a hydrophobic core. So it's all of the non-polar side chains being shoved into the middle, so a non-polar core. Okay, and then decorating the outside then are polar amino acids that can interact well with water. So we have this, I guess, this layer on the outside of amino acid side chains that can interact with water. So we have a hydrophobic core, and we have a hydrophilic outside. And this type of idea, I hope, rings a bell, because we've seen biochemical molecules that adopt this type of structure before. Remember we talked about the lipid bilayers? We talked about how we have our polar head groups arranged such that our non-polar hydrocarbon tails are all kind of packed into the middle, right? So we have our non-polar tails all packed into the core, right? So the lipid bilayer forms in a very similar way that proteins fold. We are keeping the hydrophobic parts away from water. And as it turns out, this is due to something known as the hydrophobic effect, which is very much driven by entropy and not enthalpy. This is a little bit surprising, but it boils down to water molecules being displaced. We will learn more about this in an activity that we do in class.